migrating in the Salmon River were showing signs of distress, and this included abnormal and weak swimming, somewhat like a wriggling motion through the water, and this was initially noticed by the fishermen in the stream, and they also noticed some mortality in the adult fish. And as a result, the New York State DEC initiated studies that were evaluating the health of these fish. So for initial examination, the DEC sent steelhead samples to Cornell University for physiological analysis, as well as a station in USGS for thiamine analysis. And from Cornell, they didn't have any significant findings, but as you can see from USGS, there were significant findings on the thiamine levels, with total thiamine in both the liver and the muscle samples having significant lower levels. From the Salmon River deficient steelhead, you had only 4.3 to 9.5 nanomoles per gram in the liver, and only 0.4 to 0.6 nanomoles per gram in the muscle. So why thiamine? Thiamine is a B1 vitamin that is only obtained through the diet, and it's required for proper growth, neurology, and metabolism. And there's multiple different isomers of thiamine that are abundant in an organism, and these include free thiamine, thiamine monophosphate, and thiamine pyrophosphate. And that thiamine pyrophosphate is the one that's mainly active in these organisms. So early mortality syndrome is what occurs in the offspring from adult salmonids, and this is occurring in multiple different salmonids in Lake Ontario, as well as some of the other lakes. And it's due to an insufficient maternal transfer of thiamine from the adults to the offspring. So you can see the effects during the yolk sac and first feeding stage in the offspring. And the clinical signs include loss of equilibrium, the lethargy, abnormal swimming, and often high mortality. In the image here, you can see multiple fish that are suffering from this early mortality syndrome, and as you can see, they're not doing too well. These fish are actually all still alive, but they're just lying on the bottom. And this would also increase their vulnerability to predation. So on top of the mortality that you're getting from that thiamine deficiency, you're also having additional mortality due to increased predation. And as a result, you have a lot of decreased recruitment in feral populations, and that leads to limited establishment of these salmonid populations. So following when they saw that the steelhead weren't doing too well in Salmon River, the DEC collected some of the fish at the Salmon River hatchery, and they injected them with 50 milligrams per kilogram of thiamine, and that was to improve their health for the spawning. And so the following spring, when people from our lab went to the hatchery to collect some of the samples for the analysis, they had steelhead that were kept in a pond on site that were treated with the thiamine. So we were able to collect 27 samples of the steelhead that were injected with the thiamine, and then 27 that weren't treated. And from those 54 samples, we collected egg samples as well. So they were collected for either thiamine analysis or for fertilization. And the samples that were taken for thiamine analysis were immediately frozen, and then the other samples were fertilized and either immersed in a thiamine bath at either 0 or 1,000 parts per million. And that's just another way of treating these fish for that thiamine deficiency. And Osprey mortality and the symptoms of EMS were followed throughout the development of the offspring. So this is just kind of going through the different treatments that we applied to the fish. You can see that either the adults were either treated or not treated with that injection, and then from there their eggs were taken for either thiamine analysis or fertilization. After fertilization, the eggs were either treated or untreated. So we end up with 4 different treatment groups with treated adults, treated offspring, treated adults, untreated offspring, untreated adults, treated offspring, and untreated adults, untreated offspring. And I'll be focusing on the thiamine analysis portion of this, looking at the difference in thiamine levels in the eggs for both treated adults and untreated adults. So for thiamine quantification, we use high-performance liquid chromatography, and this allows us to see the concentrations of those different isomers of thiamine. As you can see in the bottom left, there's a chromatograph that we'd likely see from using this method, and each peak represents the first peak representing the thiamine pyrophosphate, the thiamine monophosphate, and then the free thiamine, and combine those add up to equal the total thiamine in each egg sample. So here's a bar graph that's showing the differences in the thiamine isomers as well as the total thiamine between the eggs from the treated adults and untreated adults. And as you can see, there's a significant difference between the free thiamine and total thiamine among the offspring. And you can see some of the values here, the differences between them, between those egg samples. And we also noticed that there was some high standard deviation among the free thiamine and the total thiamine, and that's pretty much just showing that there were some differences in the success of the treatments. And yeah, it's pretty interesting how you had such deviation between these two. So next we'll be looking at the survival of those offspring. So this is looking at how well that injection treatment works as far as the offspring mortality. And when we think of mortality, we look at two different stages. So there's pre-hatch mortality and then there's post-hatch mortality. Pre-hatch mortality occurs from 0 to 400 degree days, and then post-hatch mortality is from 400 to 1,000 degree days. And that post-hatch mortality includes yolk sac edema, body deformities, and then mainly that EMS. So here's a scatter plot looking at the comparison between total thiamine and offspring survival for both treated and untreated groups. You can't see it too well here in the legend, but the orange circles represent the untreated group, and then the blue diamonds represent the treated group. And you can see there's a significant decrease in the post-hatch survival for the untreated group. And there's also a good portion of these offspring had 0% survival. So that's really going to impact that recruitment for that year class and possibly the whole population. So here's a couple videos comparing the treated versus untreated offspring, and hopefully it works. Yeah, okay, so you can see how these fish are swimming well throughout the water column, healthy behaviors, so the treatment is working. And then in comparison with the untreated fish, you have very abnormal behavior. They're lying on the bottom. You can see that they are still alive, and you'll also see some fish swimming spiraled when we tap the tanks to agitate them a little bit. And it just goes to show how these fish are extra vulnerable for predation and how this can really be impacting their populations. So that's just an interesting thing to look at. So now we'll be looking at a comparison among all four of the treatments to see how each treatment affects the offspring mortality. And so we looked at mortality assessment among total mortality, pre-hatch mortality, and post-hatch mortality. With the main one being post-hatch mortality, where you would have that EMS occurring. And you can see how in the untreated sample here, you have up to 85%, actually it was up to 100%, but on average 85% mortality in these offspring. And that's a significant amount of mortality for the steelhead fish when they're not treated. So just kind of makes you wonder like what could actually be happening in the lake if these fish aren't being treated outside of the hatchery. And this is based on the total number of developed eggs, and that's just because there are a good portion of eggs that weren't able to develop. So if we included those eggs, they would make these values much lower. And that's just they weren't fertilized properly, or some other factors such as that could have caused that mortality. So when looking at which treatment would work best, we found a significant difference among the survivals of all four treatments. And then we found that all treated samples, so that's the treated adults, treated offspring, untreated adults, treated offspring, and treated adults and untreated offspring, all had significantly higher percent survival than the untreated samples. And then when we were comparing the differences among each of the four samples, we found that there was no significant difference among offspring survival among those three treated groups. So that means that whether you inject them once or you have the injection as well as a treatment, like the immersion, it won't have much of a difference on the survival of the offspring. So for future projects, we're hoping to look at compare the egg thiamine levels to the muscle and liver samples from the adults that those eggs were taken from. So we can kind of see if there's a threshold level that these steelhead have where once the adults get below a certain point, the mortality starts to occur. So we can have a better idea of when this issue, the EMS issue, is occurring. And we can also evaluate the thiamine levels and EMS in other Lake Ontario salmonid species, because we do know that it's occurring in them, but we might not know information such as exact thresholds or what percentage of the offspring are dying due to this issue. And that's especially important for native species, such as the lake trout and Atlantic salmon, which are having issues with natural reproduction in the lake. So if we can see what type of, or how much recruitment they're getting based off of mortality and the survival, we might be able to have a better idea of what might be occurring in the lake and what could happen in the future. And lastly, this can help with potential causes of early mortality in salmonids. Currently, right now it's believed to be that there's a thiamine deficiency due to the enzyme thiaminase. Thiaminase pretty much just breaks down thiamine in the body and prevents it from being used. And it's very abundant in prey species such as the alewife. And there's studies that have been conducted that show a correlation between alewife abundance and thiamine deficiencies in fish. And recently in Lake Huron there was the crash in alewife. And following that crash, they noticed a significant increase in lake trout natural reproduction. Other ideas are de novo synthesis of thiaminase. So this is based on the idea that some fish are able to produce their own thiaminase. I know that with carp they're able to do this, and what that could do is just simply the fish would produce it and just pretty much wiping themselves out. It doesn't make much sense, but it's a possibility. And then lastly, prey-lipid content. This is based on an idea that was, it came up within the Baltic Sea, where there's a similar issue occurring with thiamine known as M74, and that's impacting Atlantic salmon. And they found that Atlantic salmon feeding on prey with a higher fat content had increased thiamine deficiencies. And the idea behind that is that when you have that increase in fat content in your prey, you need to oxidize that, or have antioxidants to prevent that fat from oxidizing. And thiamine may be getting used as an antioxidant, so that would decrease the reservoir for thiamine in the body and not allowing enough thiamine to be used for its natural processes, leading to that thiamine deficiency. So I'd like to thank many people for their help with this project, specifically the New York State DEC Salmon River Hatchery, for letting us use their facility and take samples and having them help us out. Andrew Noyes and Dale Honeyfield for their help and their data on the thiamine analysis, as well as laboratory assistance from many undergrads and co-graduate students, and also funding for this project was supported through the Distinguished Professors Award through the College of Brockport. Are there any questions? Yeah.
Is there any evidence that there could be natural selection on the part of the predator to, say, have a higher stomach pH or some other component that can help them, counter the effects of thiaminase, of those organisms becoming established in the gut? I guess the reason I'm asking that question is, if we prophylactically just treat, fish within the hatchery and so forth, might it be a good idea also to determine if there's an opportunity for natural selection, as well to sort of combat this issue?
I haven't seen anything as far as using other sources of treatment like that. I know that it sounds like that would be pretty specific, so we'd have to figure out more about what the exact cause of the issue is. If it were thiaminase, if it was thiaminase, then whether it's de novo synthesis or from an exterior source, it'd be interesting to see where that thiaminase is prevalent in the fish. So then we'd have to focus on what type of treatment we'd be able to do. But yeah, that'd be interesting to see, and if there's another form of treatment that we could use on the fish.
Our final talk for the morning session will be given by Alex Gack, coming to us from Hobart College, and he's speaking about mercury accumulation in lake trout.
Hi everybody, thank you for coming. My name is Alex Gash. I am an undergrad at Hobart Colleges. I'm doing my honors work right now on mercury accumulation and lake trout. I get the best of both worlds because I go to the college, but I also work at the Finger Lakes Institute part-time. So I'm part of the larger mercury project that we have going on there, looking at mercury through the food web. But today I'm just going to talk about lake trout. So lake trout are really cool. They're my favorite fish. They grow really slow, but they get really big, and they get to be really old as well. They're native to North America on the west and east coast and Canada. They are invasive in some places like Yellowstone National Park where they've been introduced. But for the most part, they are native to North America, and they prefer lithotrophic lakes where it's deep, cold, a lot of oxygen. So you're really not going to find them down south. You're going to find them up where we live. So what I'm looking at is mercury specifically, and this is important